have these two excellent scientists with me today. Lauren, who has been working with me the past 2 semesters on this project, and Deanna, who started this semester on this project. And we are doing a genomics project through Hudson Alpha Code program. Specifically, the variant they are looking at is with a gene they're interested in that is active in Alzheimer's disease. And I'll let them take it away.
Okay, so I'm Lauren White.
And I'm Deanna Snelly, and like she stated, our project is on the impact of leucine to arginine at position 282 on the PSEN1 phenotype. We use bioinformatic techniques to kind of predict how our PSEN1 mutation would have an effect on the overall structure and function of the gene that encodes for.
The clicker is not going to work because you're using your computer. Oh. Oh, working?
So for our project, like she said, we use Alzheimer's disease. Alzheimer's disease is a type of dementia that affects the memory, thinking, and behavior. Typically, people 65 years and older survive an average of 4 to 8 years, but there has been, signs that people have lived as long as 20 years. This kind of highlights how unpredictable the disease is. We try to focus in on the early onset of Alzheimer's disease because it is believed to be hereditary and through variations of specific genes such as PSEN1. PSEN1 encodes for presenilin-1 and has a genomic location of 14q24.2. Also, mutations in this gene have associations with cardiomyopathy, acne inversa, Pick's disease, as well as Alzheimer's disease. The bottom left-hand corner is the PDB file that we use and inserted into Yosara to get the presenilin-1 and the gamma secretase complex. The top red portion is actually a subunit called navcastrin. This acts as a gatekeeper and helps cleave APP and notch for presenilin-1. The left-hand subunit ATH1A and PIN2 have regulatory functions for the gamma secretase complex. In this slide, this is actually the gamma secretase complex within recognition of APP. The lower right-hand corner is turned 90 degrees to see kind of the location with it with leucine or the presenilin subunit. So presenilin APP, it cleaves off smaller peptides that have a toxic and non-toxic genes, and the more toxic ones have longer, more hydrophobic that create plaques. These plaques are found in brain scans of people who have Alzheimer's disease. These plaques are associated with brain degradation, so that's how it relates to ours. Also, this is our BLAST in the sequence alignment. So BLAST is an algorithm that uses similar genes or similar organisms compared to the ones that we're looking at. And in our case, we use humans, a mouse sequence, and a chimpanzee sequence. The sequence of the mouse and the chimpanzee were very similar to the ones that we use. And the highlighted region at the top shows like how similar it was to the ones that we use, which in further goes to what Lauren has to say.
Okay, so in this slide, we kind of took a graph where we took from ClinVar, which shows a database of all the variants of the PSEN1 gene, and we put all that into this plot between the CAT score and the amino acid position, and we were looking specifically at the uncertain variants, but you can also see here the pathogenic variants and benign. And we were looking for an uncertain variant that was in close proximity to a pathogenic variant on this graph. And as you can see, we picked the one that has the CAT score of 4 and the amino acid position 282. We thought that since it's very close to other pathogenic residues, that it probably will display a pathogenic phenotype. And so that's kind of the purpose of this graph. But as you can see that the CAT scores are very similar with 404 and 397 for the pathogenic, similar to the CATS 404 for our variant. So in this slide, you can see the variant of unknown significance that we chose was the point mutation at thymine guanine, resulting in the missense mutation from leucine arginine in position 282. So in the top image, you can see the leucine residue 282 and how close it is to the leucine residue on the APP substrate. This is important because you can see that interaction between not only the variant that we picked, but the actual substrate itself that we were concerned with. Again, the topological domain is the cytogenic region, and in the bottom right-hand corner, you can see the arginine residue and how that kind of changed in relation to the leucine residue on the 282, or on the APP. And then on this, we kind of just compared the active sites, not the active sites, the residues and the side chains of those residues. In leucine, we have more hydrocarbon side chains that makes a more hydrophobic residue as opposed to the arginine residue that's more basic in its side chain. We kind of predicted that this would affect the flexibility of the residue and that may affect the flexibility of the protein, causing conformational changes and thus changing the function of the protein itself. So that's kind of the importance of the side chains that we need to switch to. And then in this one, so we kind of compared two images that we uploaded into Yosara, showing the active sites, the active site of PSEN1, the APP substrate in blue, and the residue that we circled in red. So for the wild type residue, leucine, comparing it to the distance between the asparagine residues of the active site and the leucine residue on APP, we can see that between that and the mutant image, which is the same thing but using arginine, we saw the distance change between the arginine residue and leucine as opposed to leucine and the leucine residue in ADP. So that kind of indicated some change there, but then the pattern was reflected in the distance between the active site and arginine and the active site and leucine. So that change in distance kind of indicates a conformational change there. And that kind of backed up what we were predicting earlier. And that's kind of important because changing the active site, changing that area can change the function of that protein itself. So then lastly, we kind of put all this data into PolyPhen. PolyPhen is a software that predicts the deleterious effects of different variants, and it also predicts how pathogenic a mutation may be. It uses higher level math, but it'll break it down for you, and we kind of used, when we put this in there, the home div and home bar both had the same prediction. It was possibly damaging. That kind of backed up what we were thinking. So that's the end of our presentation, but basically we just used bioinformatic techniques to kind of predict the effect of our variant and how maybe it would cause deleterious effects on the amino acid sequence and changing the function of that protein. So, that was really good. Any questions?
Okay, I have a question. So you talked about variant selection, right? So how did you select variant in PSEN1? Like how did you decide to select that particular one?
So that goes back to the graph that we picked. We kind of took ClinVar, which is a great database that has a ton of information on the variants of pretty much any gene that you can find. So we took the entire list, and then we put it into a graph and separated it based on pathogenic, benign, and uncertain. And we were primarily just concerned with uncertain missense mutations, and you can put all that into the ClinVar database. And once we did that, once we picked it, we just kind of went from there. But we just picked an uncertain variant of unknown significance that was closely associated with a pathogenic variant in the same location, the same CAD score.
Yeah, like she said, we wanted to have it near something that was pathogenic to see if that would, it being in close relation to that, if that would have the same effect on the uncertain parts of it.
Okay, and I'm just curious, is this software from Hudson Alpha?
Hudson Alpha is actually really closely partnered with Yosara, and that's where we did all of our protein modeling, the measuring the distance between the active site and leucine residues. So that's how Hudson Alpha kind of plays in. But
Dr. Yoshida is about Hudson Alpha.
Yeah.
any other questions?
Who wants to alignment, was that for the whole gene, or was that for just a certain exon?
So we actually took, we used GenePro and we did a blast analysis of just the topological domain, the cytogenic region. And once we did that, we kind of took those results, which was like 250 different sequences that align, which I think was just that region. Then we went from there and did the aligning of those sequences. So it's just that particular region in the PSEN1 gene on each different organism.
Okay, my question is kind of, if you went back a couple of slides, there, that's the one. Okay, so your wild type and your mutant type, the leucine was hydrophobic and the arginine was hydrophilic, so I
It's
think.
basic.
But it would be more hydrophilic. And you said that would make it more flexible, I think? I guess that was where I was asking kind of what we meant by that, I guess.
So different residues, from our research, different residues have different flexibility levels. The hydrophobic, it wouldn't necessarily make it more flexible or less, or I'm not sure which one it would make it, but I'll look more into that. But I knew that certain residues do have different levels of flexibility, and changing a more basic, or changing a more hydrophobic residue to a more basic residue would impact the flexibility in some way of that residue, which may impact the flexibility of the overall protein in that region. So.
I would just, it was an interesting statement. I'm kind of curious about following up. Are those two, is that linking two separate, I'm assuming alpha chains, it looks like it's an alpha chain and something on the other side. Is that what that's doing?
Which one?
I'm looking at, well, where the circle is, where you're making the replacement, it looks like it's a bridge between two chains almost.
So I actually just circled the RG residue just so you can kind of see it.
Oh, okay.
It's a bit of a busy image, but
Okay.
basically it's just showing
Okay.
the active site.
It just looked like it was a cross bridge. And I was like, oh, okay.
But it's not a crossbridge, it just shows that residue in the back, and you can kind of, it's a little bit different on this computer, but you can kind of see the arrow of that, and that's pointing to that, pointing to that residue. But yeah.
Thank you.